So let's say that you want to introduce a foreign genetic material, a foreign sequence, into a host genome or into a nuclei of the cell or in a cytoplasm. How do you really do that? Like, what is the application, a protocol there you need to follow in order to apply this foreign genetic material into an organism? The procedure in which you introduce the foreign gene variant into a host cell is called the gene insertion, and there is the various techniques in which you can use this technology and apply this foreign material into a host cell. Into this video, I'm intending to go through the history of gene editing and insertion of genetic material. The history of gene therapy and gene editing begins in early 90s when the French Anderson introduced a healthy gene variant to a 4-year-old girl who was suffering with the SCID and immunodeficiency disorder, and the procedure was rather successful. It not only cured the child from the terrible disease, but also it showed us that the genome editing technique has rather the huge potential, which might be favorable and a game changer for future applications. So there is two main variants for the genome and gene editing techniques. First, there is the viral gene editing, and then there is the non-viral gene editing. Both of those techniques has their own pros and cons, their setbacks, and they are not really perfect right now, especially the 90s version of genome editing with viruses were really chaotic and not really reliable, because what you do with the viruses is basically you put a genome sequence into a virus and you use the virus as a code and a machinery that will deliver the genetic material to a foreign cell, to a cell that you're interested to transfect, and you don't even put genetic material into a particular section of the genome. You just dump the healthy gene or the gene of interest. So you did not have the power to put a particular sequence in a particular section of genome. You just dump the genetic material inside of that, and you hope that that genetic material would get synthesized and it would have some kind of effect on body, and you wouldn't be able to change the damaged sequence back then. That was the viral gene editing on 90s and on early 2000s. Then there is the non-viral gene editing, which is little sluggish and ineffective compared to the viral vector, because viruses are the cool tools, they are fast and they are reliable if we consider the application of adding the genes into a cell nuclei. But if we talk about the non-viral vectors, it's basically like coating genetic material with some kind of biopolymers, for example the tyrosine biopolymer that will be able to pierce right into a phospholipidous membrane of the cell and go to a nuclei and put out those genetic materials into there. So first of all, it was just dumping the genetic material into a cell nuclei or the cytoplasm, and things have changed after that. CRISPR was invented, and CRISPR made everything way easier for geneticists and molecular biologists. CRISPR consists of the three main parts in its machinery system: the guide RNA, an RNA sequence which is used to target a specific section of a genome that you want to engineer. Also, there is a template DNA, and there is the Cas9 enzyme, the most important tool in CRISPR application. The Cas9 enzyme basically swaps out guide RNA. Guide RNA goes to the particular matching sequence, and Cas9 cuts that particular sequence from an entire genome. And then the template DNA goes to that particular region and will stick to it basically. That's what Cas9 is. The application and the process is pretty straightforward, and it's one of the most inexpensive and straightforward genome editing technique that is available to research purposes and for the clinical applications. One aspect to mention is that there has been like the other more complex and hard applications before CRISPR. If we remember the TALEN and the ZFN, those were somehow similar to CRISPR but way complicated and expensive, so it was not really accessible for people, and you needed a great resources and huge amount of money to do one single experiment, especially if you do it on human. So CRISPR changed a lot of things because it's inaccessible for a lot of people, and it will make scientific progress way faster and efficient. If we consider the CRISPR viral vector application, we integrate CRISPR with the viruses. So the virus is this kind of capsule thing that can multiply and put genetic material into a host genome, and the CRISPR is the machinery which enables us to cut a particular section of DNA and insert that particular sequence of genetic material into this particular section. So you put all of those CRISPR materials, the guide RNA, the Cas9 enzyme, and the template DNA, and then you put those into a viruses in a viral vector. The most used virus for the genetic application with CRISPR is the AAV vector, because it's really reliable and it doesn't really have like the immune response to the host body. So here's my video. It's really short, but I really hope you learned something from it and it was an enjoyable experience. If you would like to see more content like this and more videos just like this, hit the like button, subscribe to my channel and share the video. Thank you and bye.